It could be Prof. Natrah already here. Good morning, Prof. Natrah.
Hi, good morning, Prof. Sabtu. Assalamualaikum. Can
Waalaikumsalam.
can you
Sorry
give
for a
me
bit earlier to
eh,
have activity. It's okay
yeah,
for you?
it's okay, yeah.
Hmm.
But can you give me just 5 minutes just to
For sure.
prepare everything?
Yeah.
All
Yeah.
right.
No
Thank
problem.
you.
Yeah, yeah. Untuk Mbak Arifah dan Mbak Nanda, tolong pastikan students sudah join semuanya ya.
Baik, Prof.
Prof. Sabtu,
Yes, yes, yes.
there seems a little bit of problem of me connecting my internet to my desktop. So I
Oh,
have to
okay.
to find a stable place to present my presentation.
Yes,
Uh
yes,
I think I need more time. Can we start
yes.
at what is the time there in Indonesia?
Yeah, it says
8
7:15.
7:15. Can we start
Yes.
at 7:30?
Yeah, 7:13. Yeah,
I'm so
7:13. Yeah.
sorry. Yeah.
Yeah, no
All
problem.
right. Okay. All right. Okay. I'll get back to you.
From Natrah, in
Uh-huh.
case there will be still a problem, you can
Uh-huh.
send the file and then we will show it for you.
But
Yeah, can be
because
like
now
that.
I'm using my phone,
Oh, all right.
so I can
Yeah,
open the presentation. I
sure,
mean, I
sure,
cannot
sure.
share through my desktop,
Yeah,
so I have to find a stable line.
no problem, no problem.
Okay.
Yeah,
All right.
waiting.
Okay. All
Okay.
right. Thank you.
Arifa, to make it easier, please allow me as a host as well, just in case there's something I can show it.
Okay, Prof. The host now is Mbak Nanda, so I will let Mbak Nanda to make Prof. Sabtu as a host.
Two, it can be three of us, yeah.
Oke, baik, Prof.
while waiting for the students complete, make sure this is recorded. Yeah, Mbak Arifan. After when we start presentation from Prof. Natrah, we start recording. Okay, Mbak Arifan.
Untuk mahasiswa dipersilakan untuk absensi terlebih dahulu menggunakan link absensi yang ada di layar monitor.
Okay. Assalamualaikum.
Waalaikumsalam.
All right, okay, I'm ready now. I'm so sorry.
No, it's a bit too early, maybe. Yeah, there. No.
Yeah, but it's okay. At least the internet is stable, so I
Yeah,
think I'm
yeah,
ready
yeah.
now. Mm.
Okay, great, great. Thank you. Here we have Ibu Arifa Rizky Nafisa. Yeah. Good morning. So this would be a house. Okay, you can actually start. Yeah, but before that, just give warm welcome lah for Prof. Natrah, my sahabat, my friend. It's been collaboration quite several years. Yeah. Okay, welcome again, and thank you so much. Yeah, for having a time for giving lecture to students. Mbak Arifah, maybe you can start it now.
Okay, baik. Thank you so much for the time, Professor Dr. Sabto Purnomo Purto, PhD, and also Professor Dr. Natrah Ihsan. Assalamualaikum warahmatullahi wabarakatuh. It's truly an honor to host of this academic forum on the global classroom, and today's session highlight the importance of sustainability marine research, particularly the role of microalgae in supporting aquaculture and human welfare, an increasingly vital topic in today's global challenge. It's my great pleasure to introduce our distinguished speaker, Professor Dr. Natrah Fatin for University Putra Malaysia, who will share her expertise and valuable insight on this topic. And also the honor to Professor Sabto Purnomo Putra PhD from Universitas Diponegoro as our moderator who will guide the session and facilitate the discussion. Before we start our UGC session, so we may take some photo first. For all the participants, my on the camera before we take the photo.
Yeah, I think for all the students to make sure that you have the camera on, please.
Okay. And
Camera
also
on,
please
please,
to
for the
Mbak
student.
Nanda to take a picture.
No, wait. We have to wait until the students also turn on the camera, please.
Okay, baik Prof. Please all the students turn on the camera to take a photo. Okay, I will start count to take a photo. 1, 2, 3 for the first layer. Okay, and the second layer. 1, 2, 3. And the third layer. Okay, 1, 2, 3. Okay, it is enough for the time to take a photo. Before we begin, allow me to inform you that this session will run from 7 to 8:40 a.m. and the participants requested to mute their microphone during the presentation, but in the last session, there will a question and answer section. Okay. Without further ado, I would like to invite our moderator, Professor Sabto Purnama Putro, PhD, to take over the session. The floor is yours, Professor. Thank you so much.
Okay, Mbak Arifah, thank you for being the host there for this valuable event. Actually, this is a part of the world class university program that launched by UNDIP this year. So I would like to express my gratitude for our speaker, Prof. Natrah Ihsan, as my colleagues, and it's been quite several years having collaboration with Prof. Natrah, and this today Prof. Natrah willing to share the expertise, especially on the algae bacteria interaction, right? So this is very very good to know and to learn, for the students, because the students actually still in a fourth semester, I think. Mbak Arifah, is the fourth semester, right? Yeah, so it's involved in the subject of marine biology. So, Prof. Natrah, without further ado, I think it's time for you to share your expertise, especially giving a lecture on the harnessing algae bacteria interaction, uh, for sustainable disease management in aquaculture and agriculture. It's very nice title, I think, actually. Okay, time is yours, Prof. Natrah.
Alright, okay. Thank you very much. Bismillahirrahmanirrahim. Assalamualaikum. Selamat pagi, my colleagues, Prof. Sabto, and also all the coordinators, Mbak Rizky, Mbak Afifah just now. Thank you for introduction. It is very nice to see you all online today, this morning. And it is my great pleasure also to, I'm very honored and also thankful to be invited to share the knowledge that I've, the knowledge through my research to all the students in UNDIP. And today I'm going to share my work, which is mostly about interaction between algae and bacteria for sustainable disease management. Specifically in aquaculture and generally also in agriculture. Okay, wait. All right. Little bit of outline of the presentation. I would first start with a little bit of introduction of bacteria. What is bacteria? Why is it important? And then how can bacteria lead to antimicrobial resistance, or also known as AMR? And how communication between bacteria or quorum sensing can play a role in terms of enhancing or making MR more serious, and how inhibiting communication between bacteria can lead to reduction, or at least as an alternative for antibiotics that can somehow reduce the occurrence of MR, and how algae can play a role as quorum sensing inhibitor in the aquatic ecosystem. All right. So let's start with a little bit of introduction of bacteria. I think most of you have know already what is bacteria. You are all already in first year. So bacteria is a very small, microscopic microorganism. They are among the oldest life form in the earth. Mostly a single cell. In terms of their role in the ecosystem, they can be neutral, very neutral. Some of them are very beneficial, but there are also bacteria that are pathogenic. Okay, however, no worries, most are actually harmless. 99% have been shown to be beneficial to human, to the environment. But we have to also be, a little bit cautious in terms of this 1% that can be harmful to us, or also known as pathogenic. So when I say pathogenic, it is harmful. Okay. Alright, so when we are talking about this 99% beneficial bacteria, they can reside in our body, okay, our colon, which basically houses around 10 to the power 14 bacteria with more than 400 species. You can also find bacteria in the fish, shrimp, intestine, and also water environment. You can also find it in the plant or also in the roots, okay, to rhizosphere. Bacteria can also act as barbieridators. So it basically can clean the environment. So it basically can also be one form of waste management. Now, when we're talking about this 1% that are harmful to us, okay, also harmful to the environment, most of these harmful bacteria, the pathogenic bacteria, they are the ones who can release toxin, okay, and this causes infection, disease, and some of them have been shown now to be resistant to antibiotic. Okay, so when they are being resistant to antibiotic, okay, they become this what we also call as superbug. Okay, superbug, they become Superman or something like that. And it has been shown that it has been predicted that by 2050, I think most of you are still alive there during that time, you are all still young. Uh, it can kill more than cases of cancer. Okay, we can see, I think, a lot of cases now, cancer, compared to before, but in 2050, there will be more cases of death that are due to superbugs or antimicrobial resistant compared to cancer itself. So it's actually a serious matter that you have to really, become aware now and also take action. Now, so what is MR? What is antimicrobial resistant? AMR is the ability of any microorganism, okay, be it bacteria, be it fungus, that can evolve and also become resistant to antimicrobial drugs such as antibiotic that used to inhibit or kill them. So remember, normally when you have fever, and that fever is basically due to bacteria. So normally, doctor will prescribe you with antibiotic, and they will also tell you, Natrah, please finish this. Okay, why do you think the doctor said that? It is because when you don't finish the antibiotic, the bacteria that reside in your body, okay, the one that is pathogenic, they can become resistant, and it's easy for them to become resistant, and also transfer their resistance to their friends, okay, through what we call as horizontal gene transfer. It's like touching their friend and then the friend become resistant. And later, when there's another time when you have surgery, for example, or you have fever that are also due to bacteria, that antibiotic wouldn't work anymore. So it is because you're not finishing your antibiotic, and your bacteria inside your body become resistant. They become superbug, and by becoming superbug, they also make their friends to become superbug, and a lot of data have shown that cases of bacteria become higher and higher. So as simple scratch, you can have your fever, but then you can also, I'm not hoping that you are dead because of MR, but that can also be causes of death, in the future, and it is more than cancer itself. So MR, this resistant microbes, you can find it in people, okay, in your body, in your cell. Okay, if your bacteria is become resistant, you can also find it in animals, in the food, and also in environment. So this is why a lot of people term it, scientists term them as one health, because when we are talking about resistant microbes, it basically covers a lot of segments. It is not only human, um all these resistant bacteria can also come from shrimp, they can also come from fish, they can also come from the water. And then when you eat your for example, the uncooked shrimp, or maybe you drink the water with this resistant bacteria, it can also be, transferred into your body, although you have finished your antibiotic. So this shows the seriousness of MR. Okay. So when we are talking about the cases of MR in aquaculture, 90% of bacteria originating in seawater have been shown to be resistant to one or more antibiotics. Okay, and up to 20% of these bacteria are resistant to at least 5 antibiotics. Okay, so that is why, when you go to the doctor, when the doctor sees that, first antibiotic didn't work, they would prescribe the second antibiotic. Then the second antibiotic didn't work, they would start to prescribe the third antibiotics. And then if all the antibiotic didn't work, so it's really hard, although you have only a simple fever, it's really hard for you to, basically be healthy again, because of this resistant bacteria. And the problem is, once the bacteria have acquired this MR gene, they may exist in the environment for a long time. So you couldn't get rid of them easily. Normally, it will stay there. And there, when it stays, it can also shape the evolution of the future resistant profile. So that is why you really have to be careful with the use of antibiotics. All right. So how do you tackle MR in aquaculture? You have to, if possible, try not to use antibiotic at all. Okay, if you have fever, I think it's still okay. But if it's just a normal fever, try to take, macam Indonesia pun dia ada banyak herba-herbanya kan, jamu-jamunya. Okay, sama juga di Malaysia. Okay, try to basically how to say treat your fever, as natural as possible. If not, then take antibiotic, but do finish it. Okay. But if suddenly there's a lot of MR cases in the environment, there's a lot of MR cases in the aquaculture system. That's why we as researchers, we have the responsibility to basically give the awareness to all the farmers outside there. Please don't use antibiotics in your aquaculture system. Okay. Remember why? Because once your bacteria becomes resistant to all of these antibiotic, okay, they wouldn't, go for a long time. They become resistant for a long time, and it can be transferred to you. Okay. And remember, the cases of death will be more than cancer itself, will be more than the maybe from COVID itself in 2050. So that's why we have to take a collective action now to say no to antibiotics. Okay, all right. So what are the alternatives? There are a lot of alternatives. One of them is probiotics, using all these beneficial microbes, or prebiotics. Prebiotic is any food or sources that can somehow stimulate the growth of probiotic. We can also use herbal medicine. Okay, we have a lot of plants that can be, that can basically treat bacteria. There are also phage therapy, or viruses that can infect and also kill bacteria. So it can basically reduce bacterial resistance. And another option is quorum quenching mechanism, that basically inhibit communication between bacteria. So it can also act one of the alternative of antibiotics. But then, what is quorum sensing? Okay, so quorum sensing is actually communication between bacteria. So like human, bacteria also communicate. And how do they communicate? It's like for me, okay, I'm communicating with you with my sound, right? Okay, but for bacteria, what they did is they can basically make their signals. They basically make their own chemical, okay, like pheromones, something like that. And then from there, they release it to the environment. And at a certain concentration, a certain threshold, they will start detecting these signal molecules, okay, or the chemical in the environment. And from there, they regulate their behavior. whether they want to become pathogenic, become harmful to the human, whether they want to become even more colorful, whether they want to produce light, something like that. Okay, they control their behavior through signal molecules or through communication. So that is why, and then there are a lot of studies that have shown that a lot of diseases in aquaculture are being controlled by this mechanism. Okay, so when they communicate, they become harmful. When they are silenced, they become less harmful, or they are not harmful at all. That's why a lot of people have been also looking at possible organism that can quench the signal molecules, that can also become quorum quenchers or also quorum sensing inhibitors. So in a way, you can decrease pathogenicity, and also in a way, you can also reduce the risk of MR just now. Alright, so this is bacteria. People always think that bacteria, they are isolated, they live alone, okay, they are mundane organisms, separate entities. But in a real scenario, bacteria is actually as active as us, as a human. No, they are not human, of course. But they are active just like us, okay. They communicate between each other, they sense, they respond to each other, they monitor the environment, and they also alter their behavior. Alright. And then how did they communicate? These are the signal molecules. Okay. Among the signal molecules are the famous one, or the most common one, is the acylated homoserine lactone, or HL. Okay. These are extracellular signal molecule, meaning that you can see here at the right hand side. Okay, you can see here the signal synthesis. This is where the bacteria basically produce the signal, okay, and from there they release it to the environment. The receptor will basically detect the concentration, and from there the certain target genes are being regulated, okay. And among the signals that are being produced by bacteria is this HL, and the HL can be found in at least 70 different gram-negative bacteria species. And there are also, certain bacteria, they only use HL, but there are also other bacteria such as Vibrio, which are also a common pathogen in aquaculture. Other than using only HL, they also use different types of signal molecules. It's like macam saya kan, saya bercakap bahasa Malaysia and juga boleh difahami oleh teman-teman serumpun saya di Indonesia kan. Sama juga Vibrio, the same with Vibrio, they have languages, okay, where they can interact with people that are between their species, all right, and they are also use specific signals that can basically being understood by other species. Okay, what are the example? Okay, just now I've told you about HL. So here the bacteria, the Vibrio, they have a type of HL known as HAI1. But at this same time, okay, they also produce another signal which is AI2, all right? And then another thing is they also also produce the third signal which is CI1. And for example, AI2 is being used for interspecies. So rather than speaking only between Vibrio, okay, AI2 is also being used to communicate with Aromonas, for example, okay. To communicate between the different Vibrio. So instead of Vibrio only, they can also communicate between Vibrio alginalyticus, Vibrio harvey, alright. So this shows that all bacteria is as intelligent as us, as human, alright. So it's like macam boleh cakap bahasa Melayu, boleh cakap bahasa Jawa, begitulah. Alright. Okay. And then these are all the common signal molecules that are being used by different bacteria. So you can see the vast, the diversity of signal molecules. Okay. So it's like bacteria also have their own world. Okay, what are the impact of them communicating? So among the impact is different genes are being regulated. Basically, the activity are based on the concentration level of these signal molecules. Genes are being switched on and off according to presence of signal molecules, and this led to certain phenotypes that are controlled by quantum sensing or or the communication between these bacteria. Alright, so you can see here blank screen, but this is a picture from the outer space where you can see at the right hand side there is this vibroluminescence, right? Okay, this is outer space. This is the Earth, and suddenly through the satellite, okay, there's an image at the sea which is as big as 250 kilometers, where they call it the Milky Seas. This is actually when researchers take sample from this Milky Sea. It shows that this Milky Sea is having a lot of different vibrios that are communicating between each other, and by this heavy communication, dia macam ada mesyuarat lah bersama all this vibrio, okay, and the signal molecules are very high, and through this communication they start to produce light, blue light, okay. So this, I'm just trying to basically convey to you the vastness of this communication, the impact that can be as big as 250 kilometers. That is big, all right. Okay, and then in terms of relevance towards aquaculture, okay, animal, okay, communication between bacteria or consenting have been shown to regulate the virulence of various aquaculture pathogens. I think you have heard about, aquaculture pathogens such as Aeromonas, Hydrophila, which is common in fishes, okay, also Salmocida in salmon, Advanced tarda, a lot of vibrios that are basically harmful to shrimp, the Streptococcus, Yersinia. These are all among the common pathogens in aquaculture, and all of these have been shown that quarantine sensing play a role in terms of their virulence. Now, and other than, when we're talking about virulence, among the studies of looking at virulence into aquatic animals, consists of, looking at quarantine regulation towards the virulence of, for example, Rubercambly in udang galah, okay, the macrobacterium, even the tiger grouper, white-leg shrimp, rotifer, artemia. So results have shown that when we put bacteria with high quarantine sensing activity, it can kill this animal, this aquaculture animal, okay. But when we have bacteria which is being mutated, okay, where we silence the bacteria, they cannot talk, okay, they cannot produce signal molecules, although they are there, okay, all these animals are safe. So this shows this important mechanism of bacterial communication that can basically lead to deaths in aquaculture animal. Alright, so that is why in our lab we continually looking at different microalgae and also its associated bacteria, whether they can interfere the communication of this pathogenic bacteria. But what makes anti-quarantine sensing different compared to antibiotic? Anti-quarantine sensing works by not killing the bacteria. Okay, they are not killing the bacteria. They are only degrading the signal molecules. They are only basically inhibiting the reception of the detection of the signal molecules in the environment. So without killing the bacteria, so it leads to lower circulatory pressure. So it means that the bacteria basically they don't feel threatened. So they are not basically they don't feel like being resistant, okay, and this would allow the host to basically let the natural immune system of themselves to take the control of their health. Compared if you kill the bacteria, it's like when someone wants to kill you, of course you are being, you feel threatened. Would you just stay quiet? Of course you will do something to maybe fight that person back, okay. So it's the same also with bacteria. Okay, if we want to kill them with antibiotic, they are becoming threatened. Okay, when they are threatened, they become resistant. Okay, that's why through anti-coron sensing, we are not killing the bacteria. We are just trying to silence them. Okay, they don't really know. The communication is, the signal molecules are being degraded. So when it's degraded, they will not, produce light, or they will not also become virulent. From there, your natural immune system will become, they are the one who will play the role. Okay. Now, there are a lot of biosensors that are available to basically measure the activity of coron sensing and also anti-coron sensing. We have here different types of biosensors, from the ones that are producing purple color to green fluorescent light, to just now from what we have seen, the luminescence. So when we provide signal molecules, they with all this coloring, okay, or all these phenotypes are being expressed. But when you have samples that can inhibit, so all this can be reduced. So from there, you can basically select, or you can basically have an idea what types of organism that can act as anti-coron sensing. Okay, so our group, we have screened quite a number of microalgae with coron chain properties. So among them is chlorella, for example, and also Thalassosera. You can see here the inhibition zone, but it's not really inhibition zone of killing the bacteria. It's actually inhibiting the color that is being regulated by coron sensing. So by having, for example, at the left hand side, we have here the disc without any inhibition or purple color inhibition. So it shows that the compound here, there's no inhibition activity. But at the right hand side, we have, with the presence of these two microalgae extract, we can see that there's this zone. So this shows that this microalgae can act as anti-coron sensing. This microalgae can basically inhibit the signal molecules of this pathogen. So other than Thalassosera, other chlorella, quite a number of common microalgae in aquaculture is also showing inhibition of coron sensing activity of bacteria, okay, of the biosensors. And other than the microalgae itself, bacteria that are associated with microalgae have also been shown to basically have the activity of coron sensing inhibitor. So this is the work of my PhD, Dr. Ain, back in 2020. So what she did is she basically isolated bacteria from chlorella, and then from there she observed that different extracts of chlorella at different growth phases basically have coron quenching activity. So you can see here all the inhibition coron sensing zone. Okay, and so with the highest activity from chlorella, for example, at late stationary. And next, what she did is she looked at interaction between chlorella and also the presence of the bacteria that are being isolated from the chlorella. What she saw that, okay, there's symbiotic activity between the both of them, where chlorella improve the growth of this coron sensing inhibition bacteria, and at the same time, the bacterium which is being isolated from chlorella also improve the growth of chlorella. So this shows that when we basically identify bacteria or algae that can really symbiotically grow together, this can be also one of the strategies to control MR in the future. All right. This is another study in 2022. This is also by another PhD of mine, where our group basically noticed that there is one strain known as Marinobacter that are very common in diatom, okay, especially in diatom, which is basically also common in the use of shrimp, okay, for shrimp. And then we also observe that specific bacteria group really reside with specific algae. So it's like microalgae having their best friend, best buddies of bacteria. So, for example, here we notice that diatom. Okay, we are working with Thalassosera, and we notice Marinobacter are among very dominant bacteria, which basically reside with diatom, and studies have shown that these bacteria, these Marinobacter, are also having the possibility of having quorum sensitive inhibition activity. All right. So yeah, it has been reported also, by other researchers that it also have anti-consensing, and among the molecules that may be responsible. Okay, when we're talking about molecules, there's still a lot of things that needs to be studies. But for the time being, there is a scientist, basically, identifying phagocytin as among the molecules that can inhibit basically can have anti-consent activity. Alright, so this is another study from our group. Because we know that it's really important to identify what is actually responsible for this inhibition activity. So we work closely with a chemist, and from there, this is actually a PLC model from LCMS data, which showed correlation activity between haloflora extracts and also quansense inhibition activity. So from here, we can see a number of potential or possible extracts. Compounds that can be responsible, but again, in order to isolate, in order to really identify it, it may take time. All right. So these are among the potential compounds that may be responsible for quorum quenching. For example, this is a type of diatom which is known as haloflora. Okay. All right. And then here we identified a lot of 10 new structures, but then we still need time to go into detail on what are the most responsible compounds that are responsible for the consent inhibition activity. Alright. So other than microalgae, our group also look at the macroalgae or seaweed, because macroalgae because bacteria are also among the most common colonizers of the surface of seaweed. All right. And then actually in research, okay, seaweed is the first one that have been shown as quorum-sense inhibitor. Okay, so the first quorum-sense inhibitor has been first detected in a type of red seaweed, which is Delicia pulchra, where scientists have also basically managed to identify the compound that is responsible for the quorum-sense inhibitor compound, which is halogenated furanone. So halogenated furanone has been shown to protect from extensive colonization by bacteria. It has also been shown to able to disrupt the HL and also multi-channel system. Remember just now we were having all this sort of communication system. So this furanone can also disrupt the multi-channel system. But at the same time, the backside of halogenated furanone is that it is toxic at high concentration. So that's why there's always a potential to bioprospect microalgae for quorum-sensitive inhibition activity. So what we did is we sampled seaweed from different places in the Peninsula Malaysia. We also sample places, for example, in Johor, where we basically take sample from the one which is near to a lot of human activities, reclamation area. We also took microalgae or seaweed sample from a healthy ecosystem. So we extract, we look at the biosensor, we did some assay, and we look at whether they have this quorum-sensitive inhibition activity. And what we observed that quite a number of seaweed also showed quorum-sensitive inhibition activity at certain concentration. And what are among the important finding that we observe also, a lot of this conservation activity can also be found in when the ecosystem is very stable. All right. Okay. Alright, so these are among the compounds that may be responsible for constant inhibition activity. Okay, and so yeah, so these are the findings. Well, what we noticed that all the microalgae, all the seaweeds, that should constant inhibition activity are normally from the healthy or undisturbed environment, while so the hypothesis is um, probably healthy seaweed would basically suppress pathogen, okay, which is basically accordance also with the finding from lamb, okay, where this researcher also found that seagrass ecosystem reduce exposure to bacterial pathogens in human, fishes, and invertebrate. Alright, so I'm ending my presentation today with a little bit of, I hope you can, a little bit of future perspective that hopefully after this would make all of you, to think more, okay, and maybe find solution together, because we know now that quantum inhibitor can basically be alternative for antibiotic, okay, one. But whether it can be used clinically or it can be used in the field, this is something that needs more and more understanding, needs more and more research, and that is why I really welcome a lot, if collaboration between different parties where we can basically look, okay, not only in Malaysia, we can also look in also Indonesia, how we can use this constant better that can also work clinically or and also in the field. Okay, another one is what is actually the most appropriate routes in terms of delivery of quantum sensing practically. Okay, for the time being, we are exploring in terms of green water system, at the diatom in the water, and see whether it can be stable. So all these pathogenic bacteria basically can be silenced in the water. All right, that is another one, the second one. And the third one is, okay, whether the impact of quantum inhibitor on the other host associated microbiota. So whether whether quantum inhibitor can really also, okay, for example, if it can protect shrimp, can it also protect tilapia, for example? All right. So whether the fourth question is whether quantum inhibitor with curative properties can also become preventive. Okay, this is something we also see more evidence, okay, coming also, where it is both curative, okay, it can cure and it can also prevent. And another thing is, although researchers always assume because it is not killing the bacteria, it would basically it will also not lead to resistance. But then there are also studies that, because bacteria is intelligent, okay, they know, okay, if we want to kill them, of course they become resistant right away. But they also know now, okay, that you want to silence them. Okay, so there are also studies that slowly that bacteria, although with quantum inhibition activity, they can become resistant at the end. All right, so it's also important to study how quick pathogen evolve resistant towards quantum inhibition agent. Some things that we can ponder together. All right. So I would like to just end my presentation by introducing my faculty. Welcome undip student, okay, researchers, all the dosen-dosen. Marilah main-main ke fakulti kami, Faculty of Agriculture. Okay, our mission is to be center of excellence in education, research and technology in tropical agriculture. I believe is the same also. For Undip, we have all 7 departments. So I'm from the Department of Aquaculture. All right. So in terms of programs, we have 7 undergraduate program and also 4 postgraduate programs. Okay. These are all the lecturers in the departments, okay, and so we have people, we have docent in the field of aquatic ecology and biology. I think some of them also give lecture to all of you, okay. So I'm in the field of live food, okay, and we also have colleagues of mine who are looking at feed technology. We have docent who are working with fish breeding and agriculture, genetic and biotechnology, and also fish health and diagnostic. All right. And other than that, I'm also the head of a research group known as ALBIC, or Algae Biotechnology and Innovation Research Group, where this is actually a combination of researchers from different fields, which cover from circular aquaculture, ecology, environment, and also engineering technology, with which basically use algae in their field. All right. And we are also active. We have our website, we have Facebook, LinkedIn, TikTok. Please so do subscribe and learn what kind of research that we are doing. And these are among the upcoming events. Actually tomorrow we would be having our focus group discussion. All right. And then Prof Sabtu will be talking here in September, yeah, Prof Sabtu. Okay. So there will be an event.
Yes.
Yeah,
Yes. Thank you for the invitation. Thank you very much.
so I'm looking
My
forward
pleasure.
to meet Prof Sabtu this September for our symposium on circular innovation in aquaculture. There is also poster session.
Yeah.
So
Yeah.
if you have if you can please do come. So the event will be on the 24th of September. So do join us. And in November we have this Algae Asia 2026, which will be organized with our industrial collaborators. Right. So I would end by thanking all my funders. I'm so grateful to IDRC who have been funding me from 2019 to look on algae as alternative of antibiotics in shrimp aquaculture, and not to forget also funding me in terms of looking at seaweed as IMTA for IMTA. All right, for IMTA, and I collaborated a lot with Prof Sabto, and we really hope that this collaboration can be for a long time, can be sustained for a long time. Okay, thank you very much.
All right. Thank you very much, Prof. Natrah, for the very nice presentation, giving lecture. It's kind of a bit heavier for the
Oh,
students,
sorry.
but this is very important.
But
But it's okay.
I hope
This
you
is
can get something.
very good to see. No, this is really needed for the students anyway, just to make sure that they also looking for the kind of modern research activities that involve sensing inhibitor. Yeah, it's kind of the like bacteria or something. Yeah,
Yeah,
that's
bacteria communicating between each other.
yeah, that to communicate to each other. Okay, that can be detected. Right. All right. So this very good content and presentation for students as well. So now time for the students to ask the question. So I just invite you, the students, to ask the question. Any students can be in Bahasa
Yeah, no
or in
problem.
English. Yeah,
Yeah,
it's
I
both of
can
them.
understand both.
Yeah, it can be both. It can be Japanese. I will transfer it into English. Okay. Yeah. All right. Please, student can ask the question by raise hand, please. Who is the first?
Oh, Yolanda?
Yeah, could be Yolanda. Yeah. Is there any other students, please? Waiting for the other students, please, Yolanda, asking the question. After that, Mbak Nadine, yeah. Who is the next after Nadine? Who is the next? I give
Okay.
a chance. It is very big opportunity for you to know and ask the question
Yeah,
regarding
you
the
can
content.
ask anything.
It can be asked anything about biology, especially microalgae and the technology as well. Okay, waiting for other students right raise hand. So please, Yolanda, ask the question, please.
Oke, oke, terima kasih untuk Prof. Sabto dan Ibu Arifah untuk kesempatannya.
You can
Perkenalkan,
actually on your camera if possible, if you don't mind.
sorry, Prof. I think my connection is not really good, so I couldn't open it.
Oh, it's okay. It's okay. Yeah. Okay. Go on.
Okay. My question is, I have one study cases. For example, a hospital use QSI to control like the microbiota infection. Is there any possibility that it's not effective it is not effective when it used for a human or even a patient, but it's effective in a laboratory? And is there any challenges when we want to using this QSI for so many aspects?
Okay.
I think.
Alright.
Thank you.
Thank you, Yolanda. Very nice
Thank you.
question. Very important too. Alright. That is true actually. From what I know, and most of the quantum sensing inhibition studies is actually at very basic or lab scale compared towards to application scale. So like in aquaculture, our first trial in terms of application is just early this year. We took a long time to make sure that it's really work in a lab scale, because it's even in lab scale it's really hard to basically make them stable. And I know for human also, I've never heard macam pil ke apa ke yang ada, okay, memang confirm yang ini ada consensing inhibition activity, so dia boleh mengelakkan penyakit. Tapi maknanya memang itulah dia, konsep dia, the fundamental knowledge is there, alright. In terms of lab evidence is there. But again, in terms of clinical trial, in terms of field trial, there are challenges, of course. In terms, okay, when I'm saying challenges, it's actually challenges in terms of stability. Kan macam kalau field trial, kadang-kadang ada resultnya, ada tak resultnya. Sebab dia kan is about inhibiting signal molecules. The best is to isolate the compound, but in order to identify, to isolate compound, it normally takes time. So kalau tanya tadi efektif ya, nampak ada buktinya, ada evidence in lab scale, but in clinical or field trial, I wouldn't say it's ready yet, but it's getting there. Okay, we have to get there. Okay, because or else we itulah nanti kan MR kan 2050 dijangka akan ada banyak resistant. So the constant inhibitor has to be ready by then. So let's work together. Terima kasih, harap menjawab.
Yeah.
Sudah, Prof. Thank you very
Is the
much.
question is answered, Yolanda?
Yes, Prof. Thank you
Okay.
very much.
Yeah, thank you. A good question. So now turn to Nadine Gaisani, and after that, Hanum. For the next, after Hanum, please raise hand. Okay. Nadine, you please.
Oke, apakah suara saya sudah terdengar?
Yes,
Okay,
clear.
dengar.
Okay, thank you in advance. Earlier you that mentioned if KSI can also affect the host normal microbiota. So how would you assess that the therapeutic benefits outweigh the risk of disrupting microbiome balance, which could trigger other disease? Like that.
Can you repeat again the question?
Okay. if KSA can also affect the host normal microbiota, how would
Okay.
you assess that the therapeutic benefits outweigh the risk of disrupting microbiome balance, which could trigger other disease?
All right. Okay. Okay. Yep. Okay. So I'm trying to answer this question based on our experience in the field. Okay. What we did is now we are now experimenting with a type of microalgae, all right, which are common in aquaculture, which is Catasaurus. And then we also isolated probiotic bacteria or cross-sensing inhibitor bacteria from this Catasaurus. And then from the isolation, we combine them again and we supply it to the shrimp. Okay. And then when we supply, okay, we use the same protocol because normally for shrimp, especially at mysis level, their first food is microalgae. Okay. And then by having this microalgae with constant inhibition activity, by having this bacteria with constant inhibition activity, we hope that there will be a microbiota balance like what you said. Okay. And then how to know? We took sample. We look at we use molecular technique, of course, metagenomic, metatranscriptomic, okay, and whether it can balance or not. Normally, in terms of balancing, okay, there are two types of organism. Okay, there are organism that are opportunists. Okay, normally the opportunists are the one which is they grow fast but they die faster. And there are also another organism that they grow slow but then they become stable. So in our case, we look at organisms that are slow, stable, and also having this constant inhibition activity. Okay, and we notice that by having this combination, okay, it would basically lead to a stable microbiota. All right. But again, whether to measure the constant inhibition activity, the level, the molecules, for the time being, we don't really have that technique yet. The most that we can do is we take back the sample, we do constant inhibition activity through to the biosensors that I've shown to you. Purple color just now, the luminescent, um, and also do like metatranscriptomic, metagenomic, just to look at the KSI gene. But that's it. But to measure the consensing molecule, we need a chemist for that. Maybe I don't know. I think HPLC, LCMS can measure that. I hope it answers.
Yes, thank you.
All right.
Okay, thank you for the next question. Yeah. Now turn to Hanum. Hanum, yeah. You can ask the question, and then following Hanum, maybe other students, please raise hand. Yeah. Okay, Hanum. Please ask the
Okay,
question.
thank you for attention. Hello, Ibu Natrah. I'm Hanum. I really interested with your presentation. Also, I interested with UPM. I hope I can pursue my study to UPM, and next month I will
Oh,
go to UPM to
fun.
do some competition.
Ah,
Hopefully I can meet you in UPM. Next, I will ask a question about quorum quenching and human stimulant. Okay. Does the effectiveness of the DC management work through nutrient competition, production of natural antimicrobial compound, of through anything in human system in cultivated organism?
in human.
Yes.
Oh, I don't really have experience in human actually. So it's really hard for me to answer. But I know for in human, I know that quantum sensing, okay, bacteria that are having this quantum sensing activity, you can find it in your teeth. The biofilm, the plaque of your teeth, okay, it has a lot of bacteria, and this bacteria basically form the biofilm through communication of quantum sensing. That one I know. And I also know that by eating a lot of garlic, turmeric, it can somehow protect your gut from this quantum sensing bacteria or quantum sensing pathogen that reside in your gut. That one I know. Okay, other than that, my knowledge is, my experience are more towards aquaculture.
Okay, thank you.
Yeah, but I hope to see you in UPM.
Yeah, actually, she will be going to Malaysia, right? For the competition, yeah.
Competition.
Yes.
Right. All the best.
What kind
Good
of
luck.
competition? What kind of competition? Tell me. Tell
Essay
us.
competition.
Essay. Oh, essay.
Yes.
Yeah. Okay, Hanum. Good luck. This is scientific competition held by UPM or what?
Bekerja sama, Pak.
Oh, in collaboration, yeah.
Yes,
With UPM.
collaboration.
Okay.
Santoso
Okay.
Foundation.
Yeah. Okay, Hanum. I wish you can meet if you can meet Prof. Natrah there, you will be very lucky because he's very busy. So,
Yeah,
but I
but yeah,
hope you
I can
can.
text.
Yeah, try to
Maybe we
go
can meet.
there. Yeah, say hi.
Okay,
Okay,
thank you,
after Prof.
Prof. and Miss Natrah.
Yeah, after Hanum, is there any students? Yolanda? Yolanda is a coordinator of this subject of marine biology. Yeah, Yolanda, any other question?
I will make sure for Prof. Give me a second, please. Thank you.
Okay. Yeah, Prof. Natrah. Seems that the students is very interested to explore more about your presentation there, and then hopefully they will be trigger the students to get engaged more about the topics, and then also maybe they come up with the idea to finish their final exams or kind of final work. Yeah, we call it scriptsy here. So this make the to inspire them. So we don't know because this still in the fourth semester. Hopefully by this regular UGC program held by UNDIP, there will be more opportunity for the students to have international experience. Then also make the students also be confident in a global community. That's the purpose actually. As I also, when I went there, I'm just impressed by the fact that the international students there also or like what we call organize the students to have passport, yeah, just to make sure that they will be going to overseas. So every students have passport. When I ask in this class, how many students have passport? Maybe only a quarter of that. So indicating that actually having international exposure is very important for the students to, yeah, at least to what we call to achieve that the university should be having international experience, both students and of course the lecturer as well. So this is the goals of this agenda, which is now set up in 2026 UGC. They call it UGC for the university global classroom, right? So this is the way that is a simple way and a cheap way to get the international exposure. Because if we send the students, let's give a lot of budget to do that. So this is a part of that program. I'm talking this waiting for your right, Sam. Yolanda. Hello, Yolanda.
Yes, Prof.
Any other
Can you
students
hear
raise
me?
hand? Yeah.
I'm so sorry, Prof. It seems like there are no further
Yeah.
questions from
Okay.
the
It's
students
free
that at
or
the
of the
moment.
student is okay, no problem. So I think this as a moderator, I will give it to the host, Mbak Arifa, to next agenda, please. Arifa.
Okay, thank you, Prof. Soto and Dr. Natrah. But I have a question actually. I'm really interested with quorum sensing, Dr. Natrah, and I want to know how can we determine that a bacterial population has reached the quorum to express the pathogenic or to express the signal? Because I really don't know how can we know this is the bacteria is reached the quorum or not. Could you tell me about that? Thank you.
All right. Okay. So in the slide just now, there's one slide where there are three pictures. One which is purple in color, another one is with green fluorescence, and the other one is with the yellow luminescence. Okay, these three are actually biosensors that you can buy in ATCC, and these biosensors basically work as biosensor where so if we have a bacteria that has quorum sensing activity and you plate it with this biosensor, it will start to produce these phenotypes that basically control the quorum sensing activity. So yeah, you need to buy the biosensor.
Okay.
Uh,
Okay, thank you for the answer about my question.
alright.
And we would to the next agenda in our forum on the global classroom. And the next agenda is Professor Dr. Sabto Purnama Putra PhD will give a certificate for Professor Dr. Natrah Fatin from University Putra Malaysia. Okay. For Miss Nanda, Ibu Hernanda Afra, maybe you can help me, okay?
Yeah. This kind of symbolizing actually now is the my expression of gratitude, yeah, for Prof. Dr. Natrah Fatin, yeah. Thank you very much for having lecture for today's under UGC Undip Global Classroom, right? So thank you very much. Thank you very much, Prof. Natrah. And this is the certificate that will be sent to you, or maybe waiting in the June when you come. We can do that both. Okay, Prof. Natrah, thank you very much.
Thank you very much. So sweet. Very nice picture. And I'm looking forward to see all of you in Malaysia and also to
Yeah,
come there soon, yeah?
sure. Yes, soon. Thank you, thank you. Yeah. Okay. So back to you, Mbak Arifah.
Okay, thank you very much for Professor Sabto Purnama Putra PhD and also Professor Dr. Natrah
Before
Fatin
we close it. Before we close it, I'm sorry. Today we have in this meeting have four lecturer actually. Prof. Natrah, I would say thank you for also Ibu Rizky as a what we call coordinator of UGC program in Department of Biology. Everything about UGC, they have to coordination with Mbak Rizky. So thank you, Mbak Rizky, again. Yeah, and also there's a lecturer as well for in the subject of marine biology, which is Mbak Hernanda Afra. Thank you, Mbak Hernanda, for gathering with us to arrange that make this event is possible. Okay, Mbak Arifah, back to you.
Okay, thank you, Prof. Safto. So this is the last in our session on the Global Classroom. We look forward to seeing you in the future version of the Undip Global Classroom, especially Professor Dr. Natrah Fatin. Thank you very much, Professor Dr. Natrah Fatin. Stay safe, stay healthy, and have a wonderful day for all participants and also Professor Sabto Purnama Putro. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam. Thank
Waalaikumsalam.
you. Bye-bye.
Bye. See
Bye.
you soon.
See you. Yeah, see you soon.
Okay,
Bye.
thank you all.